Okay, let's continue on with learning a little bit more about the cells. If you remember, the first characteristic of life is that there is biological organization, which we talked about atoms and molecules, and now we're talking about cells, and all living things are made of cells. That is the cell theory. So, hopefully you've been exposed to the different organelles at some point in time. We're going to go through them again here, and if you want, I think I have coloring pages posted out in Canvas, so make sure you go look at those. I like coloring and labeling, and I think that it helps let things stick in our heads. So there are quite a few objectives listed for this particular chapter, but really we're just talking about the different parts of both prokaryotic and eukaryotic cells. And we're going to be talking about the organelles and what they do themselves. So let's get into it. Cells are really, really diverse. And I've mentioned before, especially when talking about chapter 3 and the different macromolecules, that cells are found in all living things from bacteria to blue whales. And cells are really diverse. Even in our own body, we have a lot of different kinds of cells that are specialized for particular functions. My stomach cells secrete acid to digest my food. My eye cells receive light so that I can process that information. My neurons move that information from my eyes to my brain. And so all of those things are going to be very specialized for a particular job. We are going to talk about the generalized cells, and so we will go into each of the organelles that we might see in cells, but understand that depending on what you're looking at, you might see different structures because these cells have different functions, okay? Whether or not that's the case, most cells do require a microscope to be seen. Some of the cells that we can see with our naked eye do include things like eggs. Ostrich egg is technically a single cell, which I think is super cool. But like frog eggs, you can see with the naked eye. They're not that much smaller than an ant. And some frog eggs, like I've seen some bullfrog eggs that are huge. Some squids have neurons that are big enough that you can see with your naked eye. And so there are a lot of cells that we can see, but generally speaking, we need a light microscope to see our cells. In an online class, we have a virtual microscope that will allow you to practice the skills of using a microscope and still see images of those cells on slides. So you will get to see some of these structures even though you might be in an online class, okay? We're going to talk about bacteria as well as plant and animal cells. And again, I'm going to talk about a generalized sort of situation, but keep in mind that we are going to see a lot of variety here. Now it says most bacteria are smaller than plant and animal cells, but there have been some bacteria that have been found that are considered giants and are about the size of a fruit fly. So if you've seen fruit flies with your naked eyes, you could potentially see a particular kind of bacteria with your naked eye. And so there's just a lot of variation within this particular area of research. But let's reacquaint ourselves with the plasma membrane. If you remember, I talked about the plasma membrane when I talked about phospholipids because we have a nonpolar tail with a polar head that forms this phospholipid bilayer that is going to generate a barrier for the inside and outside of the cell. The plasma membrane is often full of carbohydrates and lipids and proteins that are playing a role in allowing that cell to regulate what goes in and out of it. It allows the cell to maintain homeostasis. It's also creating a boundary, right? If I'm talking about a yard, I might have a fence in that yard. That fence is a boundary that is going to maybe keep my dogs in the yard, but allows birds to move through the fence. Some things can move through the plasma membrane easily. Some things have to do a little bit of work or be facilitated through, which we will talk about more in chapter 5. But just keep in mind that the plasma membrane is something that we find in all cells. Okay, all cells have this plasma membrane made of a phospholipid bilayer, which is a lipid, okay? This brings us to the cell theory. I had mentioned that there are 3 unifying theories of biology: the cell theory, the theory of inheritance, and the theory of evolution. The cell theory states that all organisms are composed of cells and all cells come from pre-existing cells. And this is something that has been studied for a really long time with the same results by lots and lots of scientists. That's why it's considered a theory and not a hypothesis. for a while, before we actually had good microscopes and before we could see the cells, because we didn't see cells until the 1600s, 1700s, a lot of people thought things just spontaneously arose. So if you were to leave food out on the counter and it got infested with maggots, well, those maggots just spontaneously arose in the meat or whatever it was that was on the counter. It wasn't that the flies were laying eggs that then became maggots. It was just spontaneous. Once we had better technology, and this is the case for everything with science, when we have better technology, our understanding changes. It is updated. It is edited. And if you remember, that's a really big part of science, especially in biology. Like, we don't know everything, and we are very happy to learn something new. Like, that's one of the reasons a lot of us like science, okay? When we talk about the cell theory, we also understand that all cells have three main component parts, which is something that gives us evidence to that evolution of cells through time. All cells have that plasma membrane with the phospholipid bilayer. All cells have cytoplasm, or that jelly that's kind of inside of the cell that's holding everything like stabilized inside of the cell. And all cells have genetic material through that DNA and RNA. These three things are found in bacteria, in archaea, in plants and animals, in fungi, in protists, everybody, okay? So it doesn't matter if we're looking at prokaryotic cells or eukaryotic cells, we are going to say that all cells have these three things in common, okay? Now, prokaryotic cells lack membrane-bound nucleus. Prokaryotes include bacteria and archaea, and are going to be early in our evolutionary history. And then eukaryotic cells are your plants and animals, fungi, and protists, and they have a nucleus that houses DNA, as well as other membrane-bound organelles that are going to have specific jobs in the cell itself. That's going to be our big division in those two types of cells. So with that, we are at our first checking of our understanding. Make sure that you can answer these questions, and when you have, please continue on to the next part of the lecture. Okay, let's talk about prokaryotic cells. We can break this word down that will tell us something about these cells. Pro means before, like a prototype is your early version of a tool or a resource. So pro means before, and karyo is referring to the nucleus. So prokaryotic cells are before nucleus. So these are going to be organisms in the domains bacteria and archaea. And if you haven't heard of archaea, that's okay. They're a weird group. We'll talk about them in the diversity of life. Bacteria, though, we should be familiar with. A lot of us have heard about bacteria or even have had various bacterial infections. You have a huge microbiome of bacteria that are really important to your health and well-being. But generally speaking, anything that is considered prokaryotic is going to still have that cell membrane, it's going to have cytoplasm, and it's going to have genetic material, which includes DNA and RNA. But they are going to be very different aside from that. They are often smaller and simpler in structure than eukaryotic cells. They are considered older or more ancestral in the evolutionary lineage. And they don't have a membrane-bound nucleus. So that does not mean that their DNA is not organized. They actually have very organized circular chromosomes that are maintained within a nucleoid region of the cell, but the nucleus is a membrane that separates the DNA from the rest of the cell. Prokaryotic cells do not have that. They do have ribosomes, but ribosomes are made of RNA and are a type of genetic material. And they may have flagella. That's not going to be the same as eukaryotic flagella. Their flagella spin clockwise or counterclockwise, whereas eukaryotic flagella do the whip-like fashion. Another thing that's really unique about some prokaryotic cells, especially in bacteria, is they have a cell wall made of a polysaccharide called peptidoglycan. That is going to be a protective layer around that small cell. In any case, these guys are very unique, ubiquitous. They are considered to be living everywhere on this planet, and they're extremely successful. You know, there's no reason to change if what you're doing is working. So it's kind of one of those things, like, right? Don't change it. If it ain't broken, don't fix it. That sort of thing. Okay? We're going to dig in a little bit more to those eukaryotic cells, though, and some of those organelles within eukaryotic cells. So eukaryotic cells include any organism that has a true nucleus. U means true, and karyo refers again to that nucleus. So these are our cells that have a true nucleus. This includes the groups of protists, fungi, plantae, plants, and animals. And we're going to go through all of these different groups, bacteria, archaea, as well as our eukaryotes, in our Unit 5 Survey of the Living World. But I need you to know them here, okay? When we talk about a eukaryotic cell, we are talking about these 4 kingdoms, and they are much larger and relatively newer, okay, not as old as our prokaryotic cells. That doesn't necessarily mean that they're always larger or more complex. And one of the things that people fall into that we need to kind of break ourselves is that complexity means better. Okay, complexity does not mean better. It just means more complex, because people will have their days wrecked by some bacteria, right? I'm sure we've all had those. Okay, you get a stomach bug, you get E. coli, there are bacteria that can kill you. Like, it's absolutely a thing. And just because they're not as complex does not mean that they are less adapted or less evolved. It just means less complex. Okay, so let's talk about typical animal and plant cells, which are very similar to what we would see in protists and fungi. And so we're going to focus on plants and animals because that's what's most familiar to us as Gen Bio students. Okay, so you've probably seen this colorized version of a typical animal cell somewhere at some point in your life. I feel like every science classroom has one of these on a poster on the wall, and then they have next to it the structure of a typical plant cell. These are going to be what we normally see, and again, we're going to have various cells that are different because of the functions that they have. But when we look at a typical plant cell or a typical animal cell, we should see a couple of things in common, okay? Eukaryotic cells share an evolutionary lineage. We know this because eukaryotic cells have a lot of common structures, such as the nucleus. So you have this purple nucleus that's going to hold DNA and genetic material. The rough endoplasmic reticulum, which is studded with ribosomes, and the smooth endoplasmic reticulum. If we go back to our animal cell, same thing. Nucleus, rough ER, smooth ER. We also have a Golgi apparatus that'll have vesicles associated to it, which is membrane-bound little compartments. And we see that in the plant cell as well. Just like in our prokaryote, though, we do have that plasma membrane, the cytoplasm, as well as ribosomes and other genetic material. But we do have different organelles like mitochondria in our animal cells and mitochondria and chloroplasts in our plant cells. Yes, plants have mitochondria. Eukaryotic cells have mitochondria. It's very important that they do. Without mitochondria, there is no energy. Okay? Plant cells are a little different than animal cells because they do have that chloroplast. They also have something called a large central vacuole that is going to provide water and nutrient storage. And they have the cell membrane, the cell wall made of cellulose outside of the cell membrane. We're going to go through each of these organ systems in a little more detail and what they do. But if you can, please make sure that you can label these and then make sure that you can tell me what these different organelles do. Okay? Organelle just means little organ, and organelles are going to be membrane-bound structures that have specific functions within the cells themselves. So let's start with talking about the nucleus. Okay, the nucleus in our eukaryotic cells is a way to make sure that DNA is going to be contained in a space and stored safely, and so it can't get damaged, right? We talked about if your nucleic acids are damaged or have errors in them, it can result in proteins that are non-functional, and that can be really problematic when we look at the long-term effects of what those particular mutations can cause. So when we have our DNA, which in eukaryotes is linear, not circular like we see in our prokaryotes, we are going to have that contained within the nucleus. The DNA is only free of the nucleus when the cell is going through cellular division, okay, cellular reproduction. Ribosomes are also made of RNA. They are also still genetic material, and they are going to be found closely associated to the nucleus, as well as in the cytoplasm itself. The reason that ribosomes are so plentiful in both prokaryotic and eukaryotic cells is that ribosomes are where proteins are made. And we talked about how proteins do literally all of the work in the cells. And so we need to have lots of ribosomes so that we can make whatever proteins we need whenever we need them. Okay? Outside of the nucleus, we have something called the endomembrane system. Endo means inside, membrane obviously meaning these different little membranes around these organelles, and then a system is a connected group. So how your organ system works, for instance, right? Your digestive system starts by you chewing and then swallowing, which goes through your esophagus, which goes to your stomach, and then goes to your intestines, and each part of your body has a particular job to do. The same thing in your cells. So cells are incredibly efficient, and the reason that they're so efficient is that the work is going to be compartmentalized into these different organelles. So the endomembrane system includes the nuclear envelope, or the membrane that surrounds the nucleus, as well as the rough ER and smooth ER, different vesicles that are going to move goods from one location to another, and the Golgi apparatus. Okay. It also includes something called lysosomes, which are going to be digestive molecules. So let's go through each of these a little bit more in detail. You've probably heard of them before. This should hopefully be a refresher, but we're going to go through it anyway. Okay. So let's talk about the endoplasmic reticulum. The endoplasmic reticulum is personally one of my favorites. I used to love the rough ER and be like, man, it makes proteins, it's got ribosomes, it looks bumpy, it's super cool. It is. The rough ER is continuous with the nuclear envelope, which means that the RNA that's carrying genetic material doesn't have to go very far to get turned into proteins. The rough ER is going to take that long polypeptide chain that we talked about in chapter 3 and help it fold into that functional protein. Once that protein is made, there are going to be transport vesicles that form from the membrane of the rough ER that can then move to the Golgi apparatus to be shipped out elsewhere in either the cell itself or elsewhere in the body system of the organism that maybe you need a protein somewhere else. Okay? I learned a lot more about the smooth ER as I got to be teaching these classes, and I love the smooth ER, okay? So the smooth ER is continuous with the rough ER, but the smooth ER does different things. So where the rough ER is responsible for being a place for those ribosomes to be embedded and those proteins to be folded, the smooth ER does not have any ribosomes because the smooth ER makes lipids and carbohydrates. And specifically, the smooth ER is going to be the place that phospholipids and steroids are made, especially those hormones. But the function of the smooth ER really depends on the cell, and it can produce testosterone or it can detoxify drugs, depending on where it is in the body, which is just the coolest thing. So let's talk about this a little bit. If I look at a cell, and in that cell, I see tons of smooth ER and not a lot of anything else, there is a good chance that that cell came from a gland that is going to be responsible for producing hormones. So if I were to look at, say, an ovary cell from myself, I would expect to see a lot of smooth ER because I am in that age where I am still reproductively viable, and so I have a lot of estrogen because that's part of what my body needs to go through its cycle every month. The smooth ER plays a huge role in that. So I'm going to see tons of smooth ER in my ovary, whereas if I looked at my bicep muscle, I would not expect to find a lot of smooth ER. I would expect to find a lot more rough ER because I'm going to be needing proteins to help my muscles move more effectively. Okay? That makes sense. But the detoxifying drugs one is weird. Okay? So what happens? Well, the smooth ER is actually a place where a lot of drugs get processed and detoxified. And so if you've ever heard of someone who's built up a tolerance to something, what that likely means is their body has produced enough smooth ER in their liver cells to effectively remove that drug so that it does not have the effect. So if you, for instance, right, some people try to do like dry January. That's cool. If you drank a lot before you did that, you might start to feel better after, you kind of get used to it. And then you do the full 30 days, 31 days, you're really feeling good. You go and you're going to have a drink. And you have one glass of wine and you're like, man, I am toasted. I am such a lightweight now. What happened? Well, what happened is you stopped giving your body the signals that you needed to be able to detoxify that poison, because alcohol is technically a poison, your cells are like, oh, I don't have to do this work anymore. All right, cool. And so the rough ER and smooth ER actually can be broken down if they're not needed, just like every other organelle in the body. So when you stop giving yourself that poison, whether it's the alcohol or whether you're taking medications or whatever, your body stops producing as much smooth ER and will actually reabsorb, break it down, and reuse it in other ways. And so you become a lightweight. This is also one of the things that happens with people when they've been a heavy drug user and then got clean and had a relapse. You'll hear some statistics where people who relapse are more likely to overdose. And the reason being is that they had built up a tolerance through the regular use of whatever that drug was, and then they got clean, and they're clean for a while, and their body doesn't need it anymore, and so they've broken down that smooth ER. They're not used to that kind of damage. But because they had built up a tolerance, they feel like they need to take more of that drug. And they did when they were using it consistently. But when you stop using it and then go to use it again, you might still think you need as large of a dose as you did before. That large dose cannot be processed by the body as effectively as it was prior to getting clean, and that results in an overdose. So the endoplasmic reticulum is just this beautiful organelle that does so much work for your body, and it allows us to understand a little bit more about what goes on. It's kind of cool, right? In either case, the product from the rough ER or the smooth ER is going to be moved to the Golgi apparatus. So if my ovaries are producing estrogen, the estrogen does not stay in my ovaries. It's not the only place that it is needed in my body. It will get moved out of that cell into my blood system so it can be taken up wherever else it might need to be. The Golgi apparatus is responsible for shipping those proteins and carbohydrates and lipids to where they need to go. The Golgi apparatus is also called the Golgi body. Either way, what happens is it will receive the proteins or the carbohydrates or lipids from the ER, both the smooth and the rough ER, and it sorts and packages them for the new destination. It's making sure that wherever those products are needed, they are going to the correct place. So again, my cells might not need the estrogen that's being produced. The Golgi body will recognize that and will make sure that it leaves the cell to go out to my bloodstream. But maybe my cell has made some proteins that I need to use somewhere else. The Golgi body will still recognize that and send it out into the cell to be used. Okay? I think it's just the way that this works and how beautiful this is, and the fact that this is happening all the time without you having to think about it, you should think it's the coolest thing ever. Okay? In addition, we also have these parts of the endomembrane system called lysosomes. Lyse, if you remember, means to break. So lysosomes are organelles that are responsible for breaking things down. So when you no longer need all of that smooth ER because you stopped drinking, those lysosomes are what's responsible for breaking down those organelles that are no longer used into their parts to be reused elsewhere. Your cells are the best recyclers out there. Part of it is because the lysosomes, and part of it is just how everything is built together. Okay, these digestive enzymes are going to be responsible for even some immune responses. If you have a virus that tries to get into a cell, the lysosome might recognize that, take it up, and break it down so that your body can use it. The lysosomes are responsible for some digestion of some of the materials that your cell might pick up. But it is also responsible for breaking down anything that your cell just needs broken down. Okay? That's the endomembrane system, but that's not all of the organelles that we need to be aware of. One of the big organelles that's really useful, especially in plant cells, are vacuoles. Okay. Now, a lot of different cells will have vacuoles. Vacuoles can be used for storage. If you remember, we had starch granules and glycogen storage in our liver and our potato. That is going to be a type of vacuole. It's going to hold materials. So vacuoles are going to be sacs of membrane that can hold material. Now, they're larger than vesicles because they're going to be for storage, and sometimes they can be used to rid cells of excess water, such as what we see in the ciliate in this image A. This is a type of protist called a paramecium. It is a ciliate because it has a lot of cilia here, and it also has these vacuoles. Now, In a water environment, in an aquatic environment, water will move into cells. As that water is moving into this paramecium, it is trapping that water in a vacuole, and then it can even use this water as a form of jet propulsion to get away from predators. So that's really cool. It can help it move really quick, like an octopus would, right? Octopus do the same sort of thing, just not on the cellular level. Some of them are used for digestion, and then I mentioned how in plants they have a large central vacuole. So while some animal cells and some protists and things might have vacuoles, nothing is the same as the plant vacuole. Plants have these large open spaces if you look at a cross section of a plant cell. And sometimes it just looks empty and it's a little weird. But what it really is that is this large central vacuole that is going to be storing water. It's going to be storing nutrients in plants. It could be storing pigments. It could also be storing things like toxins. So if you've ever picked a dandelion and seen like the latex that comes out of the end of the dandelion when you pick it, that's probably coming from the vacuoles of those cells that you've disturbed by breaking those cells. So we see that in plants. And then in your fat cells, fat cells are also, we're going to see these large vacuoles, not a central vacuole like we see in the plants though. Okay. We're going to move on to everyone's favorite organelle. I usually ask people what their favorite is, and I think if I were to do an actual tally of the responses, it's like 75% of the time, the mitochondria. And they like the mitochondria because it's the powerhouse of the cell and they can remember it. All right, mitochondria are awesome. We absolutely need them, and all eukaryotic cells have mitochondria. It is really important in energy processing, which is one of those key characteristics of life, and it is responsible for taking glucose and converting that into ATP, which is the energy molecule that cells use to function. Mitochondria are unique in a slight way. So mitochondria are found in both plants and animals as well as fungi and protists. They are going to be in all of those cells. Without mitochondria, the cell does not have energy. But mitochondria is very similar to prokaryotic cells. In fact, mitochondria and chloroplasts are so similar to prokaryotic cells that they are part of something called the endosymbiotic theory. Okay, I think it's considered a theory now. It was discovered in the 1970s and proposed by Lynn Margulis that mitochondria and chloroplasts were prokaryotic cells that were absorbed by early eukaryotic cells and kept instead of digested by lysosomes. They were kept because of their capability of converting energy, which energy is so important for literally everything cells do. It makes sense that that would be a thing. Some of the different ways that that is supported is that they have their own two membranes, okay? They have their own DNA and ribosomes, and their DNA is circular, similar to what we see in those prokaryotic cells. So they do have some other similarities there as well. The mitochondria has a component part that we'll talk about a lot in playing a role in cellular respiration, but so does chloroplasts. Chloroplasts are only found in algae and plants, okay? So algae is a protist and plants are plants. And chloroplasts also are believed to be part of that endosymbiotic theory because they still have their own circular DNA and ribosomes, and they play a role in trapping sunlight and converting it to an energy source. And if you remember, energy comes into our ecosystem as solar energy and is trapped as plants and through photosynthesis, and it's eventually lost as heat by use of the mitochondria and your body doing things. So when we talk about plants, we are talking about chloroplasts and mitochondria, because while the chloroplast can make sugar, the mitochondria breaks the sugar down. Okay, we'll get into those a lot more in Unit 2. You're going to know exactly how these organelles work, and maybe mitochondria won't be your favorite anymore, but we'll find out. So check your understanding for these particular organelles. Make sure that you can tell me the component parts of the prokaryotic cell, as well as the functions of the organelles, and list all of the organelles of the eukaryotic cell. If you have any questions, please let me know. I hope you have a great rest of your day.